Great. So today we welcome Samuel Hall. He is the founder and managing partner of Scion Life Sciences, which is a venture capital firm where he's dedicated to building biotechnology companies. It includes his own wet lab space as well. So it's an interesting mix of things and how they go about devising new therapeutic ideas, whether they come from his own head or whether they're coming from outside inspiration and other scientists that he works with. So Sam started as a molecular biology student at Princeton, and upon completing his studies, he knew he kind of wanted to do what he's doing now. He had this clear vision for getting into venture capital and drug development. So he went and worked at Citigroup as a healthcare investment banker. He did that for a few years before moving to Symphony Capital, a private equity fund that's dedicated to structured investments in development level biotechnology. As that experience taught him a lot about the industry and sort of what the limitations and things are, then he opted to go and get his Ph.D. in immunology at Cambridge University in the United Kingdom. So he performed his master's and PhD studies in 4 years in Cambridge, and then came back to the States to work at Appletree Partners, where he looked at creating and building companies. And these companies, there were several of them. Syntimmune, which was acquired by Alexion Pharmaceuticals, Stoke Therapeutics, Chinook Therapeutics, Marengo Therapeutics, Ascidian Therapeutics. We're going to hear a bit about some of this work, I think, today. He served as on the board of directors for a lot of these companies. He was, involved in really the development of the therapeutics that they developed. And then as he built up this experience and expertise, he looked to branch out on his own, where he founded Scion Life Sciences, and that's where he is today. And I'm sure we're going to hear sort of the story arc and how he formulated this career path, which from the academic perspective seems a little bit nonlinear, but I think he had very set goals. And so I think that when you talk with him afterwards, really kind of figuring out what your goals are, it's a prime example of how to tailor your training to achieve them. So join me in welcoming Sam, and thank you for the people downtown that are joining us.
So you may want to mention that he's a dedicated and proud member of our
he's also I skipped over that. He's a dedicated member of our board of directors. So Sam is familiar with some of your work already from the board of directors meetings, and so please welcome.
Thank you. So you guys can hear me. Thank you, Edwin. Seems to be working. So hopefully I've got some interesting things to talk about for this group. I love what I do. I would do it for free, which I don't tell our investors, I guess, or anybody else. Unfortunately, it's just recorded and be on the internet. So probably a bad place to make that admission. But honestly, it's a lot of fun, and I think is a great way to use my scientific training. In fact, the best in terms of for me, personal satisfaction and just loving what I do. But then, as I'll get into in a couple of slides, I think my view and our strategy at Scion and the way I've done things always, is really built on the notion that in biotech entrepreneurship, in biotech venture investing, you really only do well if you solve important clinical problems. And I think that creates fundamental value for humanity and feels really good along the way when things that started, in some cases as an idea in your head or as an academic finding that you saw promise in and sprinkled fairy dust on and helped to nurture to become first a company, and then a company going into clinical development, then going into late stage clinical development, where about 5 of my companies are right now. Along the way you get to interact with patients that have been in trials of your therapy, and I think nothing's more important than helping them. And so it feels really good when you think that maybe if I was never born, this wouldn't quite have happened, at least this way. And so I titled the talk "Translating Academic Innovations into New Medicines," which is really what I do. But as Edmund said, I do want it to be interactive, and I apologize in advance because I've got a pretty busy day job and didn't have enough time to make these slides exactly as I'd like, but I think there's a lot to talk about, and we won't go in order necessarily. I'll introduce myself a little more in a second, and I think my impression is that kind of career trajectories, decisions, nodes at different places, at different points in that path are of high interest to this group, so we can talk a lot about that, but just really quick intro to Scion, and this slide actually comes from our fundraising presentation, so I apologize that some of it's out of date, but as Edmund said, I started my career as an investment banker, went to this group Symphony Capital we can talk a little bit about, and was an investor there, then unconventionally, after being in finance for a while, went back to do my Ph.D. training, and I'm really a T-cell immunologist by training. But as you'll see, I think part of my job is that I get to dive pretty deeply into and be involved in a lot of different domains of science and of medicine. And it's actually a really stimulating thing. And also, we can, in my view, we can end up quite siloed in our own fields and with prevailing techniques. And if I'm cynical about it, a certain amount of groupthink in terms of what's hot or not right now in the field and in terms of publication. And actually, the cross-pollinations between fields, I think, are an area that's really important, really stimulating and fun to kind of dip into both. Obviously, you can't be a master of all fields, but you can port over observations or techniques or approaches from one to another that actually can help to create new therapeutic opportunities. And so I've started a couple companies along that vein. But Scion is an early stage venture firm, a lot like, as I'll talk about, what I did previously at Appletree Partners. And is my own firm with my longtime partner of about 10 years, a guy called Aaron Kantoff. We were lucky to have done really well in the prior firm, Appletree Partners, where we built 7 companies that I'll talk a little bit about, or talk about some of them. That's my astonishment have gone on to generate about $10.5 billion in auditable value through being sold to larger companies or being public companies. And so that positioned us with one of the better track records out there, and that's how we were able to found Scion and raise our first fund, which is $310 million, and ended up a little bit above the high water mark indicated there of 300. But we're basically planning to do the same thing we did at Appletree, where we invested actually almost exactly the same amount, $308 million, to build those 7 companies over a period of about 7 years. And we're trying to do the same thing, build probably 6 to 7 companies out of Scion. Excitingly for me this time, and we can get into this, some of them are based on ideas that just came out of my head and the heads of our team, so we can do that. That's part of the wet lab capacity that Edmund mentioned. And we start them from scratch, like often as ideas on napkins, often as academic spin-outs, and then build them very deliberately to make sure we have high conviction of their real potential. And then kind of open the floodgates and build much more aggressively and invest much more aggressively. And I'll go through some of that. But I'm hopeful that one or more of the companies we build, over now and through the coming years, will grow up to be really important companies that are sustainably operating for a long time. And so that's what Scion does. I think along the line of career trajectories, I kept some of our team slides in here. I talked a little bit about my background, and we can talk more about that. But Aaron Kantoff is my primary partner. We've been working together for about 11 years now. Built the companies that I mentioned together, and are really complementary. I think having complementary partners that have different skill sets from you is really useful. And Aaron and I have a lot of overlapping skill sets, but he's a much better manager and operator, a little bit more of the business guy than I am. I'm a little bit more of the kind of idea guy, science guy. But we both can do everything we do and do a lot of stuff together. And then our third partner is Tad Wessel, who I met over 20 years ago. He was a couple years ahead of me at Princeton, where I did my undergrad, and then we worked together when I was an investment banker. And when I was thinking about starting Scion, I couldn't work with Aaron for a period of 2 years after our prior chapter. And so for a number of reasons, including the COVID pandemic rearing its head and not being sure what the fundraising environment would be like there, I thought it was advisable to partner with somebody and wanted that to be an old friend. And so Tad and I began working together even before Aaron could rejoin. Just in terms of our team, I'll get into what we do in more detail in a minute. By the way, how much time do I have?
About an hour.
Okay, so about 1:30, I should be wrapped up. Okay. So I thought it might be useful to you guys to understand the kinds of people that we hire into Scion. We don't have our wet lab team that Edmund mentioned here, but the core of our investment team is here. There are a couple people called venture partners or entrepreneurs in residence that is a kind of waxing and waning group I'll talk about. But Andreas, starting on the left, is a protein engineer by training. He formerly ran new technologies for Novartis, and so anything that was not a standard IgG monoclonal antibody on the biologics side coming out of Novartis, his group had a lot to do with. And so he actually was the father of chimeric antigen receptor T-cell therapies at Novartis. So that's an example of academic innovation where a guy called Carl June at Penn had actually advanced all the way through initial clinical work, the first CAR T therapies. Novartis did a deal with them to bring that in-house, and Andreas was, among others, was tasked with turning that into what became the first FDA-approved CAR T therapy. And so I connected with Andreas in the context of building a company previously at Appletree that's now called Marengo Therapeutics, which is a multi-specific antibody therapeutic company that ended up focused on cancer solely. When we started building the company, I had hoped we might do some things in autoimmunity, which is kind of my field with the technology, but we thought it was too much for the company to focus on two things and chose oncology. But Andreas, from the day I first met him in about 2015, he's a kind of a wild German guy, crazy guy, kite surfs all the time, and like he's a very talented protein engineer and therapeutic engineer, and his like quotes are "either make it and either break it" for patents. So he's a fun guy, very creative. And as you can tell, very, very much a scientist. He's actually acting as chief scientific officer of one of the companies I founded at Scion called Flux Therapeutics right now. The second person from the left is Andy Marshall. Just to give you a sense of how academic what I do can be, hired him from having been chief editor of Nature Biotech for about 20 years. And so he's got a great academic network and I think deceptively, and he's still making his transition to be an investor and is kind of daunted by that. But I as I've said to him many times and want to say to you, I think the role of being in academic publishing is actually not so very different from what I do with respect to academic sourcing, where you're looking for the most compelling therapeutic, or in the case of Nature Biotech, often technical advances in the field that are important. You go through a review process where you provide feedback to eminences, often from obviously their competitors, saying what crap their science is and how they should do their impossible experiment. But you have to interact with that academic individual and task them with doing the right experiments to show that their work really is important, which is kind of what review and follow-up experiments are, and decide essentially which translational, speaking from a Scion case and the similarity, which translational medicine projects actually have the right bone structure, merits, et cetera, to become clinically important medicines, which is really, when you're talking about translational journals, I think is what defines impact, actually. So it's actually a pretty similar role, and Andy's been great. Abhishek Datta runs the wet lab capacity that I mentioned called Scion Innovations. And he's also a protein engineer, but has had various industry roles, but very talented and has built a great team there. And then we've got, I think most relevant to this audience, have two associates right now, who are William Shore and Sam Handel. Both of them came to us. So William came in through our fellowship program, which, if some of you guys are thinking about transitioning to industry careers, there's a email list called Dropout Club. It's not the nicest name, but I think that's a little dated maybe, but historically, when I was making the transition back into industry from my Ph.D. work, that was a good source of job leads. And so so if any of you use that, you may have seen we advertised our fellowship program, which we run every year and takes postdocs, Ph.D. students, newly minted clinicians, or early career clinician scientists and clinician scientists, and gives them a chance to work with our team for somewhere between 6 and 12 months. And so William actually came to us that way. He had no prior business experience, but had just finished his MD PhD, and very smart, capable guy. And then Sam Handel joined us direct from his postdoc, where he was in the Baker lab most recently. Finished his PhD at MIT, but also had no industry experience. And so it's definitely not the case. There definitely are groups like Scion that are looking to recruit people who have, deep technical experience. And, for me, it's very important to have kind of a good, a true compass with respect to translational ideas that have the term I used before was bone structure to create important new drugs or not. So I think that's an important skill set, but it's not necessarily important to have, a finance background, et cetera. And I think that I would advise people not to be daunted by that. I think finance is a sort of specialized form of argument in my mind that is simply arithmetic. And there are some, like any new field, there are some acronyms and jargon to wrap your head around, but it's actually not that complicated. And it's something you very much can learn on the job. So I just want to go through the team, and then the rest of the talk is really structured to talk a little bit about some strategic and philosophical principles in doing what I do and how we've shaped Scion and how we invest and build companies, what our objectives are. And then I've got slides on a couple of academically originated spinouts that I've been part of that are, in one case, actually a pretty mature company going into phase 3 called Stoke Therapeutics. In another case where I've got, really just one slide, but there are some principles to talk about there. It's a company called Syntimmune that's been sold to a large pharma company. And then if we have time, I've got a series of slides on one of the companies I'm building right now called Flux Therapeutics, as well as a couple of ideas that are more nascent. But we want to make sure we use our time to talk about whatever you guys are interested in. So please, feel free to ask questions. Don't want it to be a monologue, and it's not even a well-organized monologue if it goes there. So I think, please.
Can I ask a quick question? Just, maybe that people might be interested in. So I noticed both of the people you hired as fellows look like they had some protein chemistry or biochemistry expertise. Is the idea that they will bring some competency and that you will teach them the business? What's the quid pro quo there? And do you take people who may be completely outside of what your area of interest is?
Yeah, so I mean, I think we do take people completely outside the area of interest in terms of their scientific specialization. So a former associate of mine, Chris Kane, had a different path. I mean, he came to Appletree with some industry experience, but he'd been actually a writer at a publication called BioCentury, which is one of the better and longest tenured industry rags. That rag is not a fair term to it. It's actually, they do really good journalism. And so he learned a lot there. He'd also been at another investment firm, but he studied a yeast enzyme for his Ph.D., which is, I guess maybe relevant to antifungal therapeutics potentially, but otherwise doesn't read directly onto it, notwithstanding how much yeast has taught us about genetics and lots of other things. So I don't think you have to have that one has to have scientific specialization in something that we're focused on right now, but it definitely is useful. And I think on that note, actually, Raj, one point that maybe they're thinking about for some of you is actually neither Sam nor William have animal research experience. And I think when I think about aspects of my PhD that translated and make me better at what I do now, having worked with animals is actually a big piece of that, because there are just things you don't know until you've actually, whether it's a question of how cumbersome asking a contract research organization or your team to do IV dosing of mice versus IP, unless you've done it, you don't kind of have a sense for that. And so there are things that I do where having animal work, which is almost, it's kind of binary, like whether you've done it or not, there are benefits from that. There are benefits from having a clinical perspective, but it's not, I think scientific literacy, like intellectual curiosity, the way your mind works, and kind of that nose or compass I was talking about are the most important things to me. But good question. So I was just going to frame our mission, which is a reflection of how I think about what we do as well. And so most venture firms, I don't know, is anybody thinking about going to venture or life sciences entrepreneurship broadly defined? No, okay. One person. So maybe we'll have more than one after this. We'll see. But so most of the venture industry, I may be a bit of a cynic on a lot of things, but the industry has grown up in, like many things, in a pretty haphazard, chance-driven way. And I don't know all of the fact pattern that led us to where we are, but institutional investment overall is more myopic than I think it should be. And so most all venture funds, not ours, but the vast majority, are structured as 10-year funds. So they've got 10 years to take the money that investors have entrusted to them, plow it out the door, sprinkle that fairy dust in the right places, grow the crop, harvest it, and return it to investors. And that timeline is not really compatible with how long it takes to create optimal value in early stage biotech companies that, as I said before, often start as essentially ideas on napkins. And so that's a little wrongheaded in my mind, and is accidental and drives some short-term thinking and action, where most of our peers are really in the business of taking promising translational innovations or whatever stage they get involved. And I've got a slide after this on how you create value in this space, but removing some of the risk, developing the asset, but then selling the company and the asset to large pharma and transferring the remaining risk to them, that this won't pan out to be an important drug, as well as the late stage development and commercialization, which pharma is really good at. And so they're in the business of exits. I think Scion and the way we thought about things at Appletree that I think was important in driving whatever success we've had, it's geared much more to think of our product as clinically important therapeutics. And there are a number of reasons for that, but that's a subtle difference, actually, than thinking of your objective as creating an exit. So that makes us think about, even if we are starting an idea on napkin, think through all the way to the sort of end goal of having a clinically deployed, prescribed medication that makes a big clinical difference. And it makes us think differently from a lot of peers, and select projects differently and focus differently. And so, but the objective for us, the dream, is to see, as I said, we're going to build 6 to 7 companies out of this fund. That's what we did last time with 7. And I really would love for one of them, maybe 2, if we're really good and lucky, to grow up to be a company a lot like argenx on this slide. And so what's depicted here is value creation measured by market capitalization in this emerging field of FcRn blocking therapies, which Aaron and I helped to create with our investment in Syntimmune at Appletree. And as you can see, argenx, you may have seen them advertise on TV now, which is interesting for me to see. I don't really like direct-to-consumer advertising, and I think the industry wastes a lot of money on commercial stuff. But argenx has become a $30 billion plus company on the back of this agent efgartigimod, which was a competitor of ours at Syntimmune, which is useful for the treatment of antibody-mediated autoimmune conditions. Myasthenia gravis being the biggest one for them right now, but there are a lot of IgG-mediated diseases, and they really have been underserved. And we got into building Syntimmune precisely because we thought we could build the kind of entity that argenx has become, and Scion's built to be able to, when the stars align, which is not going to be every day, but when we find an opportunity that really can go all the way like that, we are built to be able to continue investing, remain on the board, remain involved through to delivering on that. And the reason for this slide to investors is to illustrate tangibly the fact that a single clinically important therapeutic that really solves a clinical problem can generate tens of billions in value, and make you feel really good along the way. So I still remember when we ran our first proof of concept studies for Syntimmune, where we sold that company, as I mentioned before, to Alexion with kind of evolving proof of concept data in 2 indications, one of which was pemphigus vulgaris and foliaceus. And I still remember seeing the before and after photos of the first patient to go through that phase 1b study. And he's a young guy from North Carolina. I think he had, terrible, pemphigus is an autoimmune blistering disorder that was historically fatal before glucocorticoids, and results in basically 3rd-degree burns all over your body because antibodies interfere with the adhesion between neighboring keratinocytes. And this young guy, he was in his 20s, he had debilitating axial lesions where he couldn't raise his arms above that here without ripping open his scabs. And that's the before. And then after having our therapy, which interferes with the recycling of IgG and thereby reduces IgG titers, including autoantibodies that cause this disease. His lesions were totally cleared, and he could do jumping jacks. And so that feels really good when you've been involved in doing that. And that drug's gone on. But the point is, Scion's got a long-term strategy that I insisted on in structuring the fund. And instead of being a 10-year fund, we have a 22-year fund. So we can, which is a long time. And so we're really able to see things through, and that's core to how we think about things. Just a couple notes. This is a little technical, and I borrowed from a primer that I sometimes give on biotech finance. But just to give you guys a little foundation in kind of how biotech creates value for its investors, for its participants, and in addition to creating clinically important therapies. Like really what we're doing is starting with an idea at whatever stage it is, or a translational innovation, and it can be genuinely just an idea. And if you think about that, there are a lot of risks that never becomes a clinically important therapy. And those risks are why it's not worth today the same amount of money that a large pharma company would pay to get their hands on it after you've done experiments to demonstrate its value and worth. And so I've outlined here, a series kind of from discovery stage through to marketing and approval, some of those nodes. And really what you're doing is when you build a biotech company, you're taking capital, so an investment, to ask and answer a series of non-clinical / preclinical that can even be basic scientific questions, and I'll bring this to life later in the talk, that read onto the chances of actually getting to the end goal of having a clinically important therapy. And the goal is to remove more, so risk brings the valuation down. The more you remove that risk by answering these questions favorably, the more the value goes up. And your job is to run these experiments really kind of as ruthlessly as possible, because the killer experiment that really says this does or does not have value, the more binary the outcome and sort of instructive it is, the more risk you've actually removed. And so that informs a lot of how we go about building things, which I'll talk about. So given the mission that I touched on, our objective at Scion is to identify, where to sprinkle that fairy dust. We need to choose which projects to work on. And sometimes, like I said already, those are ideas that come from my brain or our team's brain. Very often they're ideas that come out of academia and labs like you guys work in. Sometimes they come out of industry, and we don't really care where they come from. I think we just want the very best ideas. And so in the process of building the companies I built before and sort of crafting our strategy, have promulgated a few what we call pillars. I think the depiction here of the Parthenon is a little trite, but my partner likes it. And but I think the ideas have a lot of merit. And so there's a lot of hype and momentum-based stuff in biotech. Things become hot, and not just like in science. I think it's not totally dissimilar. And that hype is based on some data, but maybe the data doesn't always substantiate the hype. And so in looking for translational medicine assets, projects that are really ready to make clinically important drugs today or in a tractable timeline, we focused on therapeutic modalities that are mature, meaning I often joke that in contrast to many peers, like we've never subscribed to the nucleus of the month club in the CRISPR world, where there's been, really a buoyant transmission of anything, David Liu's worked on to a new company instantly before we know whether it's actually got merit, because it's David Liu. He's a lovely guy, but it's not the case that everything that comes out of anybody's brain is great. And so there can be really overexcited translation. We don't really partake in that, and instead, I'll show a slide in a second that brings us to life, but want to work with technologies that have been through the painful process of figuring out whether you can make it. Does it have a class liability that means you could never make a clinically useful therapy out of this? What are its liabilities overall? What is it good for and not good for? And you learn that over usually 2 decades plus as these things mature. And so we tend to work with modalities that are mature, but that doesn't mean that we're not innovative at all. And so, I think that just removes important existential risks for me. And second, sounds really obvious, but most of our peers don't do this, is that we focus on diseases and drug targets and mechanisms where we can at least convince ourselves that we adequately understand what's going on to intervene, and to be able to measure in both preclinical experiments and early clinical experiments, whether you're engaging the drug target the right way, whether you're seeing changes that are highly likely to bring efficacy. And so it sounds, again, mind-numbingly obvious, but many investors and many in the biotech industry do not do this, in my opinion. And they often are distracted by an allegedly big market. Of course, your market value is zero if you haven't succeeded technically. So we really focus on technical success. And then the last of our sort of selection pillars is commercial. And if we aspire to build independent companies like Argenx that I showed you, the companies we build should have a path to doing that independently. And that's not always the case. Probably many are familiar with what's happened with the GLP-1 agonists and the obesity and metabolic disease space, as well as anybody who wants to lose weight. And that's kind of what I would call commercial sumo. So developing GLP-1 plus agonist is not the kind of thing I'd be interested in, because you've got two large entrenched companies deploying, with incredibly valuable assets for them that are spending an awful lot of money on commercial efforts and marketing these drugs, and it often goes through GPs. Like that's not a biotech tractable market, but rather when you talk about something like myasthenia gravis, where I said or pemphigus, some of the diseases we've gone after, they are rare. We're not looking for rare diseases, but they're treated by specialists that a very small sales force can go interact with and help to turn your drug into a commercial therapy. And so we want to have a line of sight to doing that. This is some of the data that I find actually quite interesting with respect to emerging modalities. It looks at over the past 50 years or so, scientific publications by year with keywords associated with each of these. And I was delighted when I saw the shape of the curves, because it's exactly the way I thought about it. And mirrors what's called the Gartner technology maturation curve. There's a consulting group that makes lots of money telling people about this curve. And if you read Wikipedia, it says that their curves are suspicious because nobody's substantiated with data. I wanted to email the company and say, I got your data. But you see, and I guess 3 instances here, at least 2. No, 3. You had Nobel Prizes awarded right in kind of the upswing of that amplification curve. You got kind of a buoyant hype phase. If you look at the dark blue, which is monoclonal antibodies, Centocor got an initial approval. I didn't think I needed this, but right around here, the first monoclonal antibody therapy from Centocor was approved. I think it caused a handful of patient deaths. And then you have a sort of valley of despair where people realize this is harder to implement than I wanted. And then you enter what's called a product generation phase for me. That's my term. But and that's where we want to get involved and invest, with a technology that's been through those growing pains and is ready to make drugs. But I find it pretty interesting how identical the kinetics of these curves are. Another kind of general principle, and Raj, this goes a little bit to your point on biologic kind of focus. So I've spent a lot of time over my career thinking about a lot of things. When I started in the therapeutic space at Symphony, we had some development partners, so some people from a contract research organization skilled in formal preclinical and clinical development that helped us with the development of the drugs that we invested in. And their attitude was that we don't understand how 95% of the pharmacopoeia works, and therefore we don't need to know how this preclinical project or discovery project we're thinking of investing in works also. And my attitude was, hold on, guys, that's not correct at all. And my conviction had long been that the better you understand the problem you're trying to solve, the better your chances of actually solving it. Which again sounds mind-numbingly obvious. There are lots of people that don't care. I always have, and this data is some of the better data to validate that viewpoint in my mind. It comes from the Biotechnology Industry Organization, and I kind of recut it, but couldn't control what the titles would be on the bars here. But it looks at about 3,000 development stage therapeutics projects and looks at their probability of advancing through each stage of development. What I'm showing here is the probability if you get into clinical trials. So phase 1 is it would be your first in human study. If you advance a therapeutic into phase 1 studies, what are the chances that it'll be ultimately approved by the FDA? And as you can see, the odds are starkly different in different spaces. And I think the reason for those differences in large measure is actually how well we understand the problems you're trying to solve, the disease you're trying to solve. So for example, cardiovascular, we understand a lot of things, but that tends to be a big, often GP or general cardiology indication where you've got heterogeneous patient populations and lots of different things going on, and you have usually historically, modest effect size or variable effects across patients. And so you see a low success rate. Psychiatry, despite all of your efforts, I think we don't really adequately understand how the brain works to intervene on things like mood disorders surgically. I think neurology for me is very different from psychiatry, but I would never invest in, and I've seen projects where it would be like, we identified that an endothelin, no offense if anybody's working on this, but we had identified that an endothelin receptor is expressed on neurons involved in pain, we think, and therefore let's develop a small molecule inhibitor of that. We have no idea what the endothelin receptor is actually doing there, et cetera. And so I literally saw a project like that and said no. And I think it ended up not working, but it didn't matter to me either way, because the chance, if you do that experiment 100 times, most of the time it's really not going to work. Lots of my peers will invest in that. Oncology, over half of venture investment and an awful lot of NIH funding, probably even more, goes into oncology. For my peers that are focused on producing exits, like I talked about, that's a perfectly good place to play, because pharma will buy development stage oncology therapeutics at high valuations all day and night. It's a big unmet need, and nothing that I'm saying is trivializing that need. But the batting average stinks compared to other places that I spend a lot more time, and it's very, very competitive, and it can be all consuming. And so we're cautious in areas that we don't understand adequately, and much more focused on those where we do. Hematology is a heterogeneous. This is non-malignant heme, but as you can see from that genetic disease, so I can go through that. So hematology, why would you have maybe a higher success rate there? Well, you're talking about the blood compartment. You can very often non-invasively take out the cells you're trying to manipulate, and measure whether you change the phenotype or the blood markers that you're trying to change, and therefore you've got a really clear idea of whether your drug's working or not early on, and often in preclinical studies. Compare that to biopsying the heart or biopsying the brain. Like it's a very different undertaking, and you generally don't do it. And so I think that's one reason that hematology probably has a higher batting average. Genetic disease is a great exemplar, because if little Johnny has 0 or 1 copies of a gene he should have 2 copies of, that his disease, the developmental component of disease may not cooperate with you, but the only thing in that context wrong with Johnny is that he doesn't have enough of this gene. And if you put it back, and you could put it back at the right time, chances are that he'd get better, because that was the single problem that caused it. Obviously, he could have other things going on, but and so for those reasons, some of these areas have higher batting averages. Long way of answering your question, Raj, on biologics. Biologics have a much higher batting average than small molecules, I think, for a lot of reasons, but one of them is that you can't design and create and optimize a monoclonal antibody against a target without knowing what the target is. Whereas with small molecules, you certainly can have a phenotypic screen that says, this cell doesn't look like it's got Alzheimer's anymore. Who knows whether that's garbage in, garbage out, or a true indicator, but you can run forward and develop something that actually you have no idea how it works. I think that's one input to that. And then last thing I'd say on this slide is selection biomarker is fascinating, right? Because that's actually an attribute of the therapies that are being measured and quantified here. And that means, you understood your target and your therapeutic undertaking well enough to promulgate a priori, I want to treat, Raj and not Diana, because Raj has a marker that Diana doesn't have. If you understand your undertaking that to that level, you've got a 26% chance of having an approved drug if you enter the clinic. That's astoundingly different from 5%. And so we try to listen to odds like this. And then, so really quickly, these are the companies that I built at AppleTree. I was going to talk to you guys about STOK in some detail and Syntimmune, but this gives a sense for you guys of the areas that I've worked in until forming Scion, and can expand on the rest of that. But they've done very well, and I think have started to validate the way that my partner Aaron and I think about the world and think about what we're trying to do. So 5 out of these 7 have advanced drugs, mostly starting from very, very earliest stages, and in several cases, mere ideas. Have advanced 5 of the 7 have advanced them into phase 3. So STOK is starting a phase 3 study. Corvidia was bought by Novo and has several phase 3s ongoing. Chinook is deep into phase 3 and ultimately was bought by Novartis. Akero is entering phase 3 for non-alcoholic steatohepatitis, which is not usually a biotech indication, but we approach that in a number of ways. And then Marengo and Ascidian are in the clinic, but in kind of phase 1, phase 1b studies. All interesting stories. Syntimmune, STOK, and Ascidian were academic spinouts, and many of the others involved sort of collaborations with academics. And so I was going to transition then to just talking about STOK, which I think Raj knows a little bit about, but I thought might be an instructive example for everyone of how this actually happens. STOK was an academic spinout from Cold Spring Harbor. Sorry, I forget. I'll give you guys background on the story first, and then a little bit about the technology and the trajectory. But so STOK was actually born out of a talk just like this that I gave at Cold Spring Harbor in 2013. And so I was invited to do like a bench to bedside, venture career thing. And I had worked with an academic called Adrian Craner, who I started the company with way back in 2006 on what became the first FDA-approved systemically administered antisense agent called nipomersin. That drug was for the treatment of hypercholesterolemia. But I got to know Adrian, who's really a splicing expert and has made important contributions to that field. Longtime professor at Cold Spring Harbor. I interacted with Adrian in the context of that because his interest had broadened to include antisense therapeutics when we were doing due diligence, and so got to know Adrian a little bit then. I gave this talk at Cold Spring Harbor and ended up chatting afterwards with a postdoc from Adrian's lab, Isabella Aznarez, and she was, very interested in starting a company. She didn't know around what, but she was agitating constantly in that direction. Set up a meeting to go meet with Adrian and Isabella and some others in the lab, and went through things that they were working on. And they had a number of antisense therapeutic efforts that all manipulated splicing. Actually, there's an important part of the story I should mention in a second, but that were, at somewhat developed stage. They had some funding. Some of them were done in collaboration with a pharma company called Ionis that was a pioneer in antisense. And most venture investors would have glommed onto those and thought about, do I invest in those or not to start a company? I think what's a little unusual about me and Scion is when ideas are testable, when there's a path to, and I'm certainly not afraid of going to things that have no data and no intellectual property. And that's where we went with this. And so the idea that we ended up building STOK around, again, was totally nascent in Adrian's lab. It had been the subject of an unfunded NIH Frontiers grant that was kind of sitting in his drawer. And I thought it was a really interesting and compelling, or potentially compelling, and certainly testable idea. And so the core of it was Adrian had been very involved for the prior 5-6 years in creating a therapy called Spinraza that some of you may know of. It really transformed the treatment of a terrible disease called spinal muscular atrophy, which is caused by having 2 dud copies, so none of SMN1. And in an accident of evolution, as again, some of you may know, I know you know, Raj, there's an essentially perfect copy of SMN1 in the genome called SMN2 that has very, very few changes, one of which is one or several base changes in an intron that results in failure to include in high abundance exon 7 in the translated or transcribed and fully spliced mRNA transcript, with a result that very little protein is produced by SMN2, but otherwise it could completely compensate for SMN1 deficiency. It really is the same protein. And so Adrian, being a splicing person, was aware of this and developed antisense therapeutics that enhanced the efficiency of that splicing event. So you did get predominant exon 7 inclusion, and therefore you had full-length transcript and translated protein that could compensate for this terrible deficiency that is fatal within the first year to 18 months of life in almost every instance. And so that had gone to Ionis, the company I mentioned, then to Biogen, and was in phase 1 trials when we started STOK. So most in the industry viewed it as unproven, but I thought it was very interesting. And Adrian's idea we built STOK around came out of that experience, and was that just as this single base or several base change that results in inefficient splicing to exon 7, inefficient inclusion in SMN2, could it be the case that a lot of genes had rate-limiting splicing events, where the majority of transcribed RNA ended up being processed in a way that was unproductive? And this is 2013. We didn't quite know that. There had been a guy called Ben Blenko in Canada, and Phil Sharp's lab had put forward a couple papers that spoke to what we were interested in. And it seemed to be a broad phenomenon, but we didn't know how many genes had this. We didn't know for sure whether we could manipulate them. But the idea was, if there are a lot of genes that have rate-limiting splicing events, we could probably use substantially the same pharmacology that was used in Spinraza to find antisense oligos that would increase the efficiency of the splicing events and drive more of the fully processed mRNA and protein product that you wanted. And so we started it as a sponsored research campaign at Cold Spring Harbor. My approach is generally to avoid. Sorry, I'm just looking at time. Okay, we're getting little low. My approach is to avoid like starting a big company and signing a huge expensive lab lease and hiring 1,000 people until you know you've got a company and project that has merit. Because I think we all want to spend our life on things that are useful and move, human health forward. And so it was great that Cold Spring Harbor let us cite the company there. We went through the process of designing a set of preclinical experiments that started in vitro to determine how many such genes were there and could we manipulate them at all ex vivo, and hired a couple lab techs to do that for the first year while we in parallel wrote patents that would underlie the company. That was all successful, and it was great to be able to do it there. In terms of what I do in my job, I was out at lab meeting every week. Like, I tend to be very involved and had, was very much a participant. Like, I'm an inventor on a number of the patents in this case, and was involved in, crafting and conducting the research campaign. We ended up showing that about 50% of humans. So the first thing we did was like $50,000 worth of RNA-seq experiments to fractionate nuclear and cytoplasmic fractions. Which is not, as you guys may be aware, routinely done, but it was very interesting. And so that analysis told us that about 50% of human genes had one or more introns that were what we called retained, meaning you could see a reasonably abundant trace for in the nucleus, but it was absent in the cytoplasm because usually those transcripts would end up being zapped by nonsense-mediated decay or other mechanisms. And so only by fractionating like that do you see this image, which is quite interesting. And so that was encouraging. And then we went about reducing it to practice, chose a number of genes with highly retained introns where we saw really abundant reads in the nucleus, but not in the cytoplasm from that intronic sequence. And kind of carpet bombed along relevant portions of that intron to see if we could find oligos that would alter the efficiency of splicing. And we made that work in a Petri dish. And that was enough for us to spin the company out and put a $5 million. I skipped over it. So depicted on what above is what we thought would happen. So the idea being that when you have these inefficient splicing events, that product is zapped when it's exported from the nucleus, or never gets out. And our objective was to increase that efflux. We called it targeted augmentation of nuclear gene output, or TANGO, because Adrian and Isabel both are from Uruguay and love tangos. So we reduced that to practice. I think that was enough to spin out the company, but ultimately actually making this work in animals, a functioning animal, proved much harder than we had hoped. And so there are always ups and downs in this kind of thing, just like in scientific research. And the first couple of targets we worked on didn't work, certainly in wild type mice. And so, sorry, I'm jumping around on this. I'll go back to those slides in a second, but you can see we founded the company with less than $1 million. This is the financing history of Stoke. Did the 5 million Series A that I was talking about. We wanted to identify a lead therapeutic, or ideally a rich pipeline of therapeutic opportunities that we could generate animal proof of concept around and wanted to take into the clinic. And that was kind of the gate in our minds for increasing our investment and really building out the company. The company was really me, a guy called Hugh Nash, who was what I call a runner, but he was a seasoned operator that acted as founding CEO in this case. and then a handful of others, but really not very many employees and not a lot. And before we built out big time, we wanted to know that there was a there there. And that took, like I said, way longer than we would have liked. We had a lot of misadventures. I think a lot of the failure that we saw was probably due to sort of auto-regulatory loops and feedback loops where in a wild type animal, if you're upregulating a dose sensitive gene, there should be ways for the animal to say either stop transcribing this or stop producing it or destroy it. And so I think those compensatory regulatory effects probably blunted our ability to see an upregulation above what would be a full wild type expression in that wild type mouse. Ultimately, we cracked the nut though with what is still Stoke's lead program, and that is in a terrible disease called Dravet syndrome. And that was a watershed moment for the company. It really helped us figure out how to practice this art broadly, which is a little harder than we hoped, obviously. But the data there were just fantastic. And a snippet from, I think, a Nature Communications paper, but it could be from a second paper in Science Translational Medicine. Sorry, I didn't have time to put the references there. Showed exactly that. So we identified SCN1A that causes Dravet syndrome. That's a haploinsufficiency, so it's not inherited. Patients have generally a de novo mutation in one copy of the gene that disables it. And so they've got 50% as much as they should. It's a perfect candidate for something where you think you can turn up the target gene in a specific manner. And so showed that this is, I think, a murine analog, but it could actually be the actual drug STK-001, showed that, as you can see on the leftmost plot, treatment it very rapidly reduced the portion of overall transcript that was in the non-productive form, meaning it still included a kind of poison exon. The productive mRNA went up in lockstep with that, obviously exactly what you want to see. And so did protein, which obviously doesn't always happen, which is why we check for RNA and protein. And so that looked beautiful. And the experiment that gave us the conviction to really build out the company in a very aggressive way, like I showed you on the financing slide, was actually this and several other preclinical efficacy experiments in the Dravet mouse, which is a good model. Actually, Dravet is a seizure disorder. I don't know if anybody's familiar with it. There aren't that many patients. You would be, Raj, but what happens is you have half as much of the sodium channel SCN1A as you should. That results in sort of underactivity of inhibitory interneurons and uncontrolled neurological activity that manifests as terrible seizures, profound intellectual disability, actually movement disorders. About 20% of these patients die before they're 15 from sudden death and epilepsy. It's really terrible. I've met a lot of patients, and it's debilitating and terrible for families. The mouse, and those seizures start around usually a child's first fever in the first year of life. So febrile seizures is a big feature of Dravet. And actually the mouse, if you put them on hot plates, gets a seizure, which is nice. They do have sudden death and epilepsy, which is the Kaplan-Meier curve on the left here. And what we showed is that when we treated the mice, virtually all of them survived, in contrast to their litter mates that all died or had to be sacked, with maybe one exception, as you can see with the 3 asterisks or 4. But that was very encouraging. We saw that this was durable. And that really formed the foundation of taking Stoke's therapy into the clinic. I'll move on in the last couple of minutes to talk about a few others and want to have questions. But these are the most recent clinical data sets from several ongoing phase 2 studies in the US and UK. And it's really gratifying to see that this therapy is working exactly as we intended, actually, in patients as well, particularly at the highest 70 mg dose group. And so Stoke is embarking on phase 3 studies. I'm hopeful that in the next couple of years, this could be an approved therapy. It's one of the ones that I'm most proud of. That's also, I think, gratifyingly really changed the way, certainly in the genetic epilepsy field, people think about therapeutic tools at their disposal. So, I mean, I go to American Epilepsy Society every year, and I'm actually working on similar developmental epileptic encephalopathies now in one of my companies, Flux. But Stoke's example and using an antisense oligo in this way has really made people think about it. And there are many efforts on many similar monogenic diseases where people are trying to research groups and clinicians are trying to practice a similar art. So it's really nice to see that you're inspiring the field like that. I think some of them will be hard, but
How did you guys determine the dose?
yeah. So that's a good question. As you probably know, Raj, that's a core question. I mean, there's so much in what I do that is important and bears talking about it in obviously limited time. But so I think dose escalation studies are really important clinically, are a really important part of drug development where you want to, it's a common mistake for people to take forward the wrong dose. It's either inactive or too toxic, sometimes both. And so getting the dose right is very important to me. We did a series of dose ranging studies, of which like these phase 2 studies are one. And STK01 was very interesting. And this is another like peculiar lesson, but before you choose your clinical dose, as you know, Raj, you do preclinical studies to convince the FDA and other regulators that it's safe enough to go into humans and there's enough chance of efficacy to merit putting patients in harm's way. And so our toxicology studies for STK01 initially in non-human primates and rats showed no tox at all. You couldn't find any dose-responsive tox, which sounds like a good thing. But the FDA is not comforted by that. They want to know what to watch for in the clinic. And so we actually had to rerun our tox studies. These are hundreds of thousands to millions of dollars studies. So this is not small potatoes. And it was a big delay for us. And our teams decided, given that we didn't escalate far enough to see any tox in the first studies, let's not make that same mistake again. And so we blew the dose out at the top, right? And the result was actually, we saw really no dose responsive tox between nothing and having to sack the non-human primate, probably because of osmotic effects, because we were putting so much fluid into this is an intrathecal drug, putting so much in there. And so we saw hind limb weakness and other things that probably have nothing to do with the therapy, but rather the administration. And that made the FDA tell us to go slowly. And so it was actually a longer path than we thought. That's a long answer on a simple question. So I guess just looking at the clock, I can stop there and just talk about whatever you guys are interested in. I've got a bunch more slides and stories, but hopefully that's somewhat interesting. And I thought STOK was definitely a salient one where it's run more of its natural history from kind of birth or conception through to becoming a real company that's a public company now. It's worth about $1 billion or 850, 900. It's been worth double that before, but it's got a line of sight to doing exactly what I wanted to do, which is I did not include data here. Most development in Dravet has focused on seizure activity, and that's the approvable endpoint. STOK has just released data over the past few months. It's the first therapeutic approach to show actual changes in the developmental trajectory of these patients, where very few of them ever are verbal. Many of them are never toilet trained. They are profoundly disabled, and across all measures of neurodevelopment, we've seen really good progress, including in actually pretty mature patients that are over 18. And so that's really encouraging. And that's actually what we want to do, is take these patients who don't have a perfect life ahead of them and get them on a more normal trajectory. So I'll be proud if we do that.
So for STOK, they're kind of hitting the check marks that tell you could take the company to the next level. But if a company isn't getting those check marks, I'm sure the scientists who poured their time and effort to these experiments might be a little bit hesitant to just give up on the project. But how do you balance, troubleshooting versus knowing when to just pull out of it?
That's a great question. That's really core to what we do. So there are a lot of answers to that. One is just your mindset. I think I would argue, and I said it at the beginning, I think most of us want to spend our life and our blood, sweat, and tears on important things. And if you're blowing on embers, that's actually not important, right? So I think, it is, there's constantly a tension between, and that's central to my job is, should we cut bait or keep going? And science is not easy. It does not follow usually a totally linear path with no surprises. And so you need to be able to, take those setbacks and obstacles and challenges, in stride, but also be very honest about them. And if you've come to the edge of a cliff, stop walking, right? Like you don't want to go off that. I've got a serious responsibility to our investors who've given us, over $300 million to do these things with. and really, it would be unfortunate and maybe criminal for me to put their good money after bad. So we need to make that decision. And I really had a bunch of sleepless nights in 2016 and 2017 on exactly that issue with Stoke, because we were getting close to $10 million invested. The idea was great. I think had a lot of potential, but we just had not seen more than about 15 to 20% upregulation for any of the targets we worked on in vivo. And thankfully, the Dravet data that I showed you came along at a point where we kind of needed a win, but that's a tension in every case. I think one way that I've gone about systematically addressing that is actually with Scion Innovations, our in-house development, our wet lab capacity, because one reason that I think a lot of biotech companies continue blowing on embers, which I think is not a good use of time, resources, money, attention, is because they're actually employed by the company. It's very, very hard to ruthlessly do the killer experiment if you think I'm not going to be able to pay my rent next month if this experiment doesn't go well, or I'm not going to be able to keep my kids in school or buy them their new shark backpack or whatever. And so I've tried to very consciously take that out of the equation by having a talented wet lab team at Innovations that can do the early. So it's kind of, I put this box there to demarcate, where we had conviction. Before that, our approach is to have Scion Innovations lead a lot of the work, not to have anybody employed by the company, not even incorporate a company, because we want to kill ruthlessly if that's what the data says, and not have precocious and premature emotional attachment. Not everyone does that at all, for both good and bad reasons, but it's a great question. I don't know if I answered. Thanks, Beth.
Other questions in the room? Or on Zoom?
Bye, Raj. Thanks.
If you are developing the question, if you are developing an idea based on a rare disease, like let's say myasthenia gravis, where it's diagnosed later in the life and where the target population is smaller, how much once it has crossed that bar, because of failure of investment, will this go down? Because if it's a rare disease, the investment might drop down later after innovation, right?
Drop down later? I'm not sure I understand.
Even if you have enough compelling data that the drug is useful.
Oh, you're saying you might decide that the opportunity is too small, even though you were scientifically successful. Yeah, so that's a challenge. I think mostly what we do, and what the pharmaceutical industry does to a fault probably, is focus on opportunities that are bigger. So there really are some orphan drugs. Perhaps paradoxically, though, or against what you would expect, people like me have been plowing money into, quote, people view ultra orphan as being really good orphan, not incredibly rare. And so we've been studying and working on diseases that I think don't really hit a realistic commercial threshold. But my approach is that filters up front for us, right? So it's sad to say, but also not, because the commercial opportunity really isn't, in most respects, intrinsically linked to the scale of the clinical need, right? Like if there are 5 people with a disease, it takes about the same amount of time to develop a drug for them. I'd rather spend my time and resources developing a drug that helps tens of thousands of people than 5, right? I mean, I'm very sorry for the 5, but you do need to make these decisions. So we make that up front and basically look for diseases that have at least a few thousand patients in the US, which, believe it or not, it can work. Myasthenia gravis is not too small at all. I think it's when you're talking about hundreds to 1,000 patients, depending on what kind of therapy you're developing. But we would not invest in the work to do all that unless we thought that success would be rewarding for our investors, basically.
So does this influence what question you will pick up at the very beginning also?
Yeah, I mean, so what you're pointing to is what I call dry lab questions. So, I mean, those are things diligence you need to learn about a disease and a clinical scenario, standard of care, et cetera. So we definitely spend a lot of time working on that, and that is an input to whether it's worth doing the wet lab work.
Thank you.
Okay, other questions? Kathleen?
Thank you for the. My question is, about drug prices.
Drug prices?
So, like, yes.
Yeah.
So,
I'm glad you asked. I'd love to talk about that.
yeah, the first, I mean, I know of some people
Mhm.
on like cancer meds,
Mhm.
they're like $20,000 a month.
Out of their own pocket?
Some,
Okay.
and like Spinraza is, I mean, all right. So like, how is, how do you, in your whole investment, journey, how do you keep prices that are realistic for
Yeah.
people to actually
I think,
use the drugs?
yeah, I think I understand. I'm really glad you asked. I meant to say something about that because it's something I care a lot about, not because I want to get rich, but as you can tell from my comment on some of the marketing commercial activity of pharma, I think there's a lot of waste and drug prices are almost certainly too high in many instances. I've got very little patience for kind of price gouging. There are a bunch of what are called specialty pharma companies that take, in many cases, non-innovative drugs that have been around for a long time, but have only a few producers and jack up the price. That all gets a lot of attention, but is not. And I think it's very bad behavior, and I hate it because people like me get painted with the brush that people are using to paint, these criminals. But the reality, I mean, I'm very frustrated doing what I do. Most people who do what I do have kind of the attitude that I have, and really are in it because they want to help patients and use their skills, talents, education to do so. There aren't that many that I've encountered kind of big conspiracy theories, et cetera. I think there is some bad behavior in large pharma, and I think the commercial guys are pretty wasteful. But I think I'm very frustrated by how much attention we give to drug pricing politically. I think the industry has made some serious missteps in how we talk about it. That if I had all the time in the world, I'd love to help fix because I think it's not good for society. But the reality is that drug prices may or may not be too high. I think you need to talk about different instances. It's been framed by the industry as we need a lot of money because we spend so much money to develop these things. I think that's flawed logic because just because something costs you a lot of money doesn't mean it was worth doing or worth what you're asking for. It's to me, a much better approach is to think about things the way I do, which is when you solve a clinically important problem that's damaging people's lives, shortening their lives, having collateral effects on their loved ones, that has value to society and to those patients. And I think a pharma company that creates an innovation that alleviates all of that suffering and problem should get rightfully, and it's not wrong to ask for a chunk of the value that's been created. Not obviously not all, otherwise there's nothing. But in America, we really politicize drug prices. I think because we don't have that much savings and copays are actually really high, and that's actually an underinsurance problem, right? So like if we have insurance, your therapeutic benefits should be covered, and when you get a prescription drug, your doctor has decided that you actually need this. There's no what's called moral hazard in the economic sense where, to me, it's sensible to have copays on a doctor's visit because you can have people who are hypochondriac and go every 10 minutes and that burdens the system and you want to disincentivize that. There's no such risk on prescription drugs, right? Your doctor has said, Diana needs this thing for her health for this reason, and they've written a prescription for it and handed it. So like there's no, but insurance companies have essentially shifted an inappropriate amount of the cost of drugs onto the patient. And most Americans blame pharma companies for that. The price of a drug has nothing to do with your copay, actually, really. I mean, it has a little bit to do because the insurers are more likely to ask for a high copay on drugs they don't want them to pay for themselves. But that's burdened patients with this. And I don't know what the solutions are to it other than I think it should be fine to have high-priced drugs when you're doing something like Spinraza does, take infants that were certain to die, and there are 6 to 10-year-olds walking around talking, which had never in human history ever happened to a spinal muscular atrophy patient before that. I don't know what that's worth, but saving a life is, if we pay people $1 million for spilling coffee on their lap in a lawsuit, it's probably worth $1 million or $2 million to have a baby having a normal life that would otherwise be dead. So like I have that view. My real problem is, does anybody know what percentage of national healthcare spending is on prescription drugs? Any guesses? Please give me a guess.
10%.
That's an unfortunate guess because that's actually almost exactly right. It was 11% in 2022. I was actually fishing for a guess that I usually get like 40 to 70% is the typical view. But my issue with the pricing debate is that it is the highest figure I've ever seen. It varies by year depending on what's going on. The highest figure I've seen is about 17%, but 10 to 13% is more typical. We could put every pharma company out of business and taking the 11%, you'd still have 95% of this same health spending problem. And so it's actually not a solution to our problems as a country. And so I'm frustrated to see that get all the attention when there's no possibility of a solution, either practically or empirically.
I think part of what drives that debate is that in other countries,
yeah,
the cost of the exact same drug is significantly less.
Right.
So either that means that, our 6 to 8-year-olds that are walking around with SMA are much more valuable, or that the industry has taken advantage of a system that is, a lot more free market here, or has maximized their profits based on what's acceptable in the different countries, right, which is a political question and is a regulatory question.
Yeah,
right?
No, I think that's right. I think Americans, obviously, as probably all of us are aware, pay much higher prices for drugs, and we're the dominant global market from a pharma perspective. But I don't know exactly, and I've read a lot on it. I don't know exactly how we got here, but mostly what's actually happened, in my understanding, is many of the ex-US systems have actually abdicated their responsibility or declined to pay a high price. So when I develop a new therapy, the US is a really important driver of that because it is more lucrative. And, I think the US didn't ask to do that. And it's not that the countries, mostly with central, I think it's accidental because they had central payers that had better negotiating leverage. But the fact, what really has happened is they've abdicated to pay their share more so than the US has decided to pay more. And I think it's hard to figure out how to fix it because I think you're not going to get Europe to pay. I think long-term what I expect, and my whole strategy is kind of based on this. I think we're going to gravitate towards, but not quite get to a European pricing system in this country. And when I look at new projects, I look through the lens of, would this work if we had dramatic price cuts? And all my stuff would, because we're ideally saving lives. I think what becomes non-viable if pricing pressure becomes much, is actually results in reform, would be kind of silly innovations that don't come with health economic value, cosmetic therapies, things that are very incremental. They shouldn't get a high price actually. But so I've solved that by going after big problems that are usually severe or life-threatening. And I think when somebody was going to die and they don't die because of something you did, like people will pay you something that is worthwhile no matter what. It's kind of my philosophy.
Other questions? I guess I had one that you kind of touched on, like with Stoke, there came a period where you kind of needed that critical experiment. So, and it sounds like an amplification of what happens in academia as well, right? There's pressure to produce whatever the target is, a novel therapy, or in this case, something that hits the target that leads to potential data misrepresentation, fraud. How do you suss out those things? Because it happens throughout science, right? It happens in the best academic labs, right? We've seen it topple, giants in academia.
Yeah.
It happens in industry. How do you, make sure that what you're getting at Scion, that what you're getting is actually the ground truth for the experiments, and is that replication in your lab, in independent places, or how does that work?
Yeah, I think so. Repetition is one thing. I mean, so Scion Innovations is able to do that and look for that. I think some investors say that they do that as like a matter of course. We don't. It's not one size fits all. Like I didn't go through flux, but do I have it in here? No, sorry, I don't have the data in here. But we founded that company out of a guy called Henry Coldcraft's lab at Columbia. He's an ion channel guy. And there was some initial data in cystic fibrosis. It was published in Nature Methods subsequent to us working on it. Like, you know, for academic in vivo experiment, Scott Canner, the MD PhD student who did the work, had, you know, 12 to 15 mice in each group or cultures in each group. I mean, seeing a decent number of data points as opposed to 3 with like a t-test on it. I mean, there's an element of trust, but your question is a really good one, Edwin, and I think about it a lot, probably much more than most investors. And your use of STOK to ask the question is very apt, because I still remember early days of STOK when we were still at Cold Spring Harbor. You know, we would periodically present the state of our work to my colleagues at Appletree. And I had the gang in from Cold Spring Harbor, and I was editing the slides before going through them with my partners. And I moved a Western blot from one side to the other side of the slide, and one of the bands didn't come. And I was like, okay, not how I'm used to seeing it. So this tech, we'd hired a couple techs for the company. I was always mindful of not putting any pressure on these individuals, but nonetheless, for reasons I still can't understand, this woman felt that she should forge her data to show the progress that she was, you know, ideally hoped to achieve, but obviously hadn't in that instance. We fired her immediately. So like, definitely don't do that anywhere, whether it's industry or academic. But I never forgot it. And that's part of why we've got Scion Innovations and all the things I said about it being really hard to ruthlessly run that experiment, the killer experiment, if you don't know whether you can buy your kid the like Paw Patrol backpack they need if it fails. It applies equally to fraud, right? Like, I think it's hard to design and run that experiment the right way. It's also hard to faithfully report the results of it if those are unfortunate and put you out of a job. And so I've dealt with that partly by putting a lot of that early sausage making into Scion Innovations rather than a company where people think I won't be employed anymore if this data looks bad, because I think that's a dangerous place to be, basically. But you know, I think it weighs on us a lot.
Okay.
And by the way, I mean, I think academic malpractice and research fraud has terrible implications wherever you are. But we're talking about putting, you know, I invested $91 million in building STOK, and the others invested several $100 million in it eventually over time. And so, like, you're actually putting a lot of eggs in a basket that doesn't deserve them in what I do if your data isn't accurate, that you're making that decision based on. So it's really important.
Thank you very much. Let's give Sam a warm thank you.